Okay, so this unit here talks about eukaryotic organisms. Of course, we're going to focus more on the eukaryotic microorganisms. But then the second part of this will be talking about the non-cellular pathogens. Non-cellular pathogens means basically viruses, prions, and virioids. All right, so that's the tail end of the, or second half of the unit. Okay. All right. I don't know what's going on. So the first slide that we go over, hopefully you've seen this, huh?
No, the device is not turning.
Yeah, I know. So, I'll deal with it. All right. Thanks for ruining my video. No, it's okay. Just kidding. All right. So the first slide that I'm going to talk about is endosymbiosis. This you should have heard about in your Gen Bio. If not, no worries. I'm about to talk about it. Endosymbiosis, or also referred to as the endosymbiont theory, it is a theory. Why? Because no one was there to see it happening, but it is a theory, but it's a solid theory. Yeah, good luck trying to disprove this one. Now, the endosymbiont theory is trying to explain, it's a theory that tries to explain, where did we get our mitochondria from? That's the question. Where did my mitochondria come from? Did I always have a mitochondria in the cell? Or did the cell, the ancestral cell, now that's what we're talking about. I know where I got my mitochondria from my mom. So did you, from your mom, not my mom, right? So I know that, but where did the first eukaryotic cell get its mitochondria from? That was a very good question, actually. So the theory is that the proto-eukaryote did not have membrane-bound organelles like we have now, but through adaptation, evolution, it started developing these internal membranes. Then now this would be referred to as the eukaryotic cell or the proto-eukaryotic cell. It had the nucleus, it had some Golgi apparatus, they said endoplasmic reticulum, those kinds. So it started developing internal membranes. It looks a lot like the eukaryotes of today. The only difference here is that it doesn't have a mitochondria, not yet. So around that time, you got to understand something. It was a bacterial world before the eukaryotes came on the scene. And we did that in Unit 1. We showed you that for billions of years, all it was is bacteria, and then after that, archaea, for quite a long time. And then this guy comes on the scene, and then develops internal membranes, and then says, I want to explore. I want to do new things. Life as a single cell is so boring. Now the problem now is for this cell to become multicellular, to develop new things, including you, it needed ATP. And it needed a lot of ATP. The ATP that this one is producing is not enough. It is not enough to become multicellular. It is not enough to develop a digestive system, a nervous system, a brain, a heart that beats for your entire life, demanding ATP. It's not enough. So it looks next door, and it looks around its environment. It realizes something. This little thing over there, called bacteria, it's very good at making ATP. It is a professional at making ATP. Yeah, because it's been around for a while, you see? So he says, you know what? Let me see if I can make a deal. Hey, bacteria, I got an offer for you. What? come live inside. Live inside, endo. In harmony with me, symbiosis. And I'll offer you anything you want. I'll give you anything you want. You want food? I got it. You want protection? I got it. You want your pH balanced? I got it. But one thing you need to do for me. What is that? Make ATP. Sounds like a good deal to me. You'll give me everything you want? I want? Yes. And all I have to do is pay back with ATP. Yes, good. So this bacteria entered the cell, the proto-eukaryote, and lived in harmony within the cell. That's this endosymbiosis, if you break it down. Fast forward many years. It became your mitochondria. Now, you can't take that back out. You can't kick it back out. All right, go. Live on your own now. It won't live. It can't. It got too accustomed to living inside the cell. It's not bacteria anymore. It is now a mitochondria. It can do things on its own, some things, but it still needs that relationship. So one of the things it can do on its own, watch this. It can replicate inside your cytoplasm all on its own. You mean you don't need to have the cell itself replicate for the mitochondria to not replicate? That's exactly what I mean. Your cell itself can replicate, that's fine, but the replication of the mitochondria is independent of the replication of the cell. Every time you go lift weights, you're stimulating your cells, not necessary to replicate. You're lifting weight to get them to grow in size, but then it realizes, I need ATP. This guy is actually serious about lifting weights. He actually got off the couch. I need to help him out. It starts making a mitochondria. So the mitochondria can replicate within your cytoplasm, independent of the cell replicating. The mitochondria does other things, all on its own. Protein synthesis. It can carry out protein synthesis within the mitochondria, independent of the cell. We'll talk about it. So, based on what I just said, it kind of looks like it looks like an organism living within your cell. That's what it looks like. Yeah, because that's what it was. What was it? Bacteria. And what did it become? Your mitochondria. So I hope you agree with me. If the theory, for me to support this theory, right? If I dive into your mitochondria, I will find a lot of things about it that is similar to bacteria. Do you agree? That's what I would think. If I dive in and I find it, oh, it doesn't look anything like bacteria, then that theory is out. So we're going to find out. Are there things in the mitochondria that are exactly like what we see in bacteria of today? What do you think the answer is? Yes, of course. And I'll share some of those with you. Yeah. So, fast forward, this cell, of course, now has a plenty of supply of ATP because of now the mitochondria, and it started exploring. It started producing new organisms, and all the eukaryotes that you see, including you, came from this cell. All the eukaryotes that are not photosynthetic, all the eukaryotes that are non-photosynthetic came from this cell. So this would be referred to as heterotrophic eukaryotes. Okay.
Those are the non-photosynthetic.
Non-photosynthetic, yeah.
Hetero.
Came from here. Like us, for example. I wish I was photosynthetic. Save a lot of money, but I'm not. So this cell realized something else. What are these green bacteria doing here? Because there were other bacteria that were photosynthetic, that are photosynthetic, not were, are, they still exist. They're called cyanobacteria. So it says, what are they doing? Oh, look at them making what? Glucose. Oxygen is a waste product of photosynthesis. Photosynthesis' main product is glucose, sugar. And just like the previous cell, it was very jealous of that too. I wish I could do that. Oh, I remember I did this. Hey, come in here, become my mitochondria. I'll give everything that you want, right? Yeah. And he accepted. Yeah, well, let me try. It now struck a deal with the photosynthetic bacteria, cyanobacteria. And it says, hey, come in here. I'll give you everything you want. Yeah, on one condition. What? Make glucose for me. Please remember that the product of photosynthesis, the goal of photosynthesis, is to make sugar. Are you making oxygen? Yes, but it's garbage. The photosynthetic cell kicks it out. Garbage. Exactly like your CO2 is garbage of cellular respiration. So this guy says, looks like a good deal to me. Went in there, lived, adapted, evolved, and became the, what did it become? The chloroplast. All eukaryotic photosynthetic organisms came from here. And what would they be? Eukaryotic photosynthetic. Plants. Algae. There we go. Nice. Plants and algae. That's it. So now the question is, how solid is this theory? Because nobody was there. Well, I will say it's solid only if I go into the mitochondria and the chloroplast now and find things that are exactly like bacteria. Do you agree? Good. Let me show you some, or share some of these things with you. The mitochondria has DNA. The mitochondria has its own DNA, independent of the DNA in your nucleus. The DNA in your nucleus, they look like shoelaces. They're linear. They're called chromosomes. The DNA in the mitochondria, that should not be a surprise to you. It's circular. Why? Because we did unit 2. What is the shape of the chromosome in bacteria? Is it linear or is it circular? It's circular. Yeah. Yeah, so the mitochondria has circular DNA. And it's the only place you have circular DNA. Because that was not part of you to begin with. What was it before? Bacteria. So some scientists may say, yeah, just a random chance. I mean, it just happens to be that. Okay, if I tell you other things, would it now help you support this theory? Yeah. How about one more thing? Yeah, maybe. The ribosome. The ribosomes outside here and the ones made in the nucleus, those are ADS ribosomes. That's their size, ATS. When you put them together, the large subunit and the small subunit together, they combine to a size of ADS. The ribosomes in your mitochondria are 70S. Again, when you combine them together, the large and the small subunit, you get 70S. What do you think the size of the bacterial ribosome is?
70S?
70S. So far I gave you 2 supporting evidence. Number 1, what did I say about the chromosome? Circular.
Circular.
Just like
Like bacteria?
Bacteria. What did I say about the ribosome? Size?
ATS.
Ribosome?
The ribosome is
70.
7, yes. Just like
Like bacteria.
bacteria.
And what is, I'm sorry, what is ATS?
ADS is the ribosome out here,
From the eukaryotic.
the eukaryotic ones.
The eukaryotic ribosomes.
The only place you have a 70S ribosome is in your mitochondria. Let me ask you this question. What if you were a plant cell? Where would you have 70S ribosomes?
If you're a plant cell,
If you're a plant cell, where would you have 70S ribosomes?
the chloroplast,
In the chloroplast.
because they used to be bacteria.
Yeah, but what else? And the mitochondria.
Yeah.
You know, this is my way of telling you something that you may not have learned properly. I get a lot of students, I don't know where they get this thing from. Then they swear up and down that their professor told them this. Some of them say, my high school teacher told me. And what is that? The chloroplast, yeah, replaces the mitochondria. I go, oh, really? No, it doesn't. They think that if you have a chloroplast, that replaces what the mitochondria does. No, it doesn't. The mitochondria is for cellular respiration. The main purpose of mitochondria is to make the energy molecule. What is that one? ATP. Very good. What was the main purpose of the chloroplast again? To make
Glucose or sugar.
glucose. So how could they be the same? How could they substitute for each other? If one makes glucose and the other one makes ATP. So if I was not here today, I would bring a microbiology substitute. One that can actually teach this stuff. If I bring in a teacher, an English teacher, can't substitute for me. And I can't substitute for him. We teach two different things, right?
Mhm.
So just because of the fact that the mitochondria does something, that the chloroplast does something completely, they can't be replacing each other. I always give this analogy to students. It's like you foolishly telling me that my gas pedal replaces my brake pedal on the car. Don't try that. Don't try it. You're gonna hurt yourself. Just because they're both pedals does not mean they do the same thing. You see? So I gotta correct that. I don't know where you hear it from, but you hear students tell me this, and I think I know what the problem is. The ATP. They heard that the chloroplast makes ATP, and that is true. So they think, well, then it can replace the mitochondria. They didn't hear the rest of the story. Okay, the chloroplast makes ATP, yeah. For the purpose of making the glucose. The little bit of ATP the chloroplast makes, it uses to make the glucose. My analogy of that is that if I had $5 in my pocket, and I go to this machine over there, it's very expensive Starbucks coffee there. How much does it cost? About $5, let's say. I'm using the money as a way to get to the sugar. Do you see what I mean? So after I go to the machine, what will I have in my hand? The $5 or the sugar? Yeah. It's exactly that way. So the students hear half the story, but they don't hear the full story. The chloroplast does not replace the mitochondria. Listen to me now. All eukaryotic cells, or at least a majority of them, have a mitochondria. The photosynthetic ones will have a mitochondria and a chloroplast. That's the key word. What was the key word? And. Not instead of. And. All right. So let me review now. 70S ribosome, circular DNA. Oh, I know one more. How it replicates. Yeah, I just told you that. Didn't I tell you this mitochondria can replicate independent of the cell?
Yep.
Guess how it replicates? Say it.
Circle?
Say it. Binary
Binary
fission.
fission, they used to be bacteria.
Oh, it's amazing. It's the only place in your whole body that does binary fission. Because it used to be.
Bacteria.
Yeah. I was helping my son studying biology. Now, when I was learning this stuff, I don't think I ever read into that, but I actually read into it in his book, 9th grade AP biology, and I was like, oh, that's pretty interesting. I didn't know that. The cytoskeleton protein. The cytoskeleton proteins that you have here, which is basically your cytoskeleton proteins, are different from the cytoskeleton proteins inside the mitochondria. What do you think the cytoskeleton proteins inside the mitochondria resemble? The cytoskeleton proteins for or of bacteria. So I already gave you four supporting evidence. I'll give you more later when we start talking about gene expression and things like that. More later. All right, any questions on this? All right. So, of course, all the other cellular structures and how we compare them. Well, this one is showing you a comparison between the mitochondria, the bacteria, and the chloroplast. That's exactly what I was just talking about. So this is all the justifications, size, and oh, here's another one, electron transport. Watch this. This is important to get because we're gonna talk about cellular respiration. The electron transport location in the mitochondria, it's in the inner membrane. If you remember from cellular respiration, complex 1, complex 2, 3, and 4, that's in the inner membrane of the mitochondria. But it's in the cell membrane of bacteria. The same exact machinery. Nothing different about it. Same exact one. Because if it's working, don't break it, right? Same exact one. Is in the cell membrane of bacteria. Now let's think about it. The cell membrane of bacteria, the inner membrane of the mitochondria was the cell membrane of bacteria. You see? Before the bacteria was engulfed, the cell membrane of bacteria became the inner membrane of your mitochondria. So if you think about it, here it is. Here's the cell that engulfed the bacteria. It's about to engulf it. Sounds good?
Mm-hmm.
This is the cell membrane of bacteria before it gets engulfed. That's where the machinery is, the electron transport chain. Sounds good so far? Now fast forward. Here it is after it was engulfed. Watch. This is still the cell membrane of the bacteria, but what is this one? This is your cell membrane.
Mm-hmm.
Well, the proto-eukaryote cell membrane. So the proto-eukaryote cell membrane became the outer membrane of the mitochondria. The cell membrane of the bacteria became the inner membrane of the mitochondria. Same thing with the chloroplast. That's another additional support. Besides the circular DNA, the 70S ribosome, how it duplicates, you see, binary fission just like bacteria. Here's another one. The fact that you have multiple layers around the mitochondria. The mitochondria doesn't have one membrane, it has two membranes. Its size, very similar in size. Sensitivity to antibiotics. Oh, that's important. Hang on one second. That's important, sensitivity to antibiotics. When you take an antibiotic, some of the antibiotics that we take are designed to target the 70S ribosome of bacteria. An example of that is tetracycline. Tetracycline targets the bacterial ribosome. And there's no bacteria that can survive with an inactivated ribosome. No way. Because what does the ribosome do? Makes proteins. And definitely, all bacteria will need a functioning ribosome. But you don't want them around because they're causing you illness. You're sick. So you take a tetracycline. It goes and binds to the 70S ribosome of bacteria. It's very selective. It doesn't touch your 80S. They go. It goes directly to this one, inhibits it, and the bacteria dies. There's a problem with that. If you take too much of it, you can potentially get hurt by it. Why? Who's talking? Go ahead.
It's attaching to the ones in your mitochondria, so you can't be
It's going to attack the ones in the mitochondria. Turning off what?
protein synthesis.
Okay, but turning off what for you? What's the mitochondria doing?
It's radiating ATP,
What's the name of that process?
cellular respiration.
There you go. It turns off cellular respiration, potentially. You may not survive that. Because everything about you screams what? I need ATP. That's a problem. But I gotta be careful. When it says use as directed by your physician, yeah, do that. Because they know if you take too much of it, I know it targets the 70S, but hopefully they know that that your doctor knows that there's a 70S ribosome in your mitochondria that can actually get hurt by that too. And same idea for chloroplasts as well. Again, they have 70S ribosomes too. All right, so these are the supporting evidence, and we'll do more as we get into other topics. All right, the domain eukarya. Oh, look at this. Whole bunch of examples. Under Animalia, the only thing that we really worry about in this class are the helminths, the parasitic worms. We don't do plants because they're not microscopic and they're not going to be part of this class here. So we're going to focus mainly on protista, which are subdivided into algae, the photosynthetic algae, and the protozoans like amoeba, for example, paramecium. We'll talk about that. And we'll also talk about fungi like yeast and other things as well. I'll tell you something about our resources, Dr. Patil and I. All of these pictures are our pictures. None of them are from the internet or anything like that. And I was making this one, and I'm like, where do I find a mushroom? I wanted a picture. And I said, wait a minute, let me take my phone out, walk in the backyard, and I found one. Just one. I took a picture of it, and the weirdest thing is, as soon as I took a picture of it, it died. Look at that, it sacrificed its life for you guys, right? And that was weird. Now, my wife is sitting in the kitchen, what is he doing? I do some weird things, right? But I got it. All right. But all of these pictures, they are pretty much ours there. All right. So let's talk about these kind of things. Again, the organelles, that's on you. It's a laundry list of things that each one of them does its own thing. We talked specifically about the mitochondria and the chloroplast, but you need to get into a little bit more and know exactly what each one of these two do, and then we will get into the detail of these things later. But I'll see how I can challenge you with this. All right, so here all they are. Here are all of them: the lysosome, the Golgi apparatus, the cell membrane, chloroplast, and all that. So make sure you know all of this, all the organelles. All right, then the endomembrane system. Either today or Tuesday, depends on the timing. One of you will teach this. Most likely Tuesday. Sounds good? Because I want to go over the organelles first, or I want to have you guys go over the organelles first, and then cap it with this one. Because this one is basically showing you how multiple organelles come together to work together to traffic proteins. So the endomembrane system's main job is protein trafficking. So there's nothing illegal going on. It's just proteins, right? What does that mean, protein trafficking? That means basically making them, right?
To deliver.
And delivering them. That's what protein trafficking is. Delivering them where? Well, it depends on which protein you're talking about.
Yeah.
Some of the proteins that this endomembrane system makes are destined to leave the cell.
Yeah. The membrane.
They're called secreted proteins. So whoever it is who's going to teach this needs to tell me that. Needs to tell me how this thing works. And you also need to tell me which proteins does the endomembrane system make. So write it down so that when you maybe go home and watch a YouTube video, you have a kind of like a heads up. What proteins does the endomembrane system make? Secreted proteins. That's one of them, actually.
That's one of them.
One kind.
It makes 3.
Yeah, exactly. So one is secreted proteins like what? Like hormones.
Insulin.
Like insulin. Yeah. Insulin has to leave the cell. So any protein that leaves the cell, there it is. The endomembrane system is making it. The other one
Membrane.
is the membrane proteins.
That's how
Yeah.
they're inside.
The proteins that are designed or destined to live within the membrane. They're called membrane proteins. What's an example of that? Oh, you name it. Receptors,
Insulin
sodium
receptor,
channel, calcium channel, transporters,
Protein.
aquaporin.
Yeah.
Any protein that's found in the cell membrane is made by the endomembrane system. I'll give you again the example, a couple of examples. Sodium channel, receptors, those kind of things. And the third kind are hydrolytic enzymes. Yeah. And where are they going to be found, Jonas?
Lysosomes.
Lysosomes. So the endomembrane system makes hydrolytic enzymes, enzymes that digest your macromolecules.
Yeah.
I can tell you something. You don't want to slip and fall into a pool of hydrolytic enzymes. You're not coming out of that pool. You're going to be part of the pool. Dissolved. Gone. They're very powerful enzymes. That's why you have to put them in a container called the
Lysosome.
lysosome.
The recycle.
Yeah. If the lysosome leaks out its contents, and on top of that, the cytoplasm is a little or slightly acidic, oh, that cell is dead. Luckily, our cytoplasm is not acidic, so even if it does leak out, those enzymes don't work. The enzymes work under acidic conditions. Thank God. It's a protection kind of thing. In case they leak out, this is not acidic, so they're like, okay, I can't do my job anyways. But if it happens to be acidic, for whatever reason, and this guy leaks out, oh, you are in trouble. That cell is gone. So these hydrolytic enzymes work under acidic conditions. So they're put in a container, a lysosome container. What do you think the pH is in there? Basic, neutral, or acidic? Acidic. And when you study this a little further in your studies, you're going to learn that there's actually a proton pump pumping the hydrogen ions in there to drop the pH, to make sure it stays acidic. So we call that your recycling center. If your cell wants to degrade a dead mitochondria, it's the lysosome that's going to do this. Here's the dead mitochondria. The lysosome will attach to it and degrade it. Does anyone know what that's called from Gen Bio?
What?
When you're recycling dead organelles using the lysosome.
Through the cytoplasm?
Yeah, in the cytoplasm. You have a dead mitochondria. The lysosome attaches to it, spills its contents, and degrades the dead mitochondria. What is the name of that? Anyone know? We mention it one time and then we just move on. Maybe that's not fair because you only hear it once. Have you ever heard of autophagy?
Autophagy, yeah.
Yeah, I know you heard it. I know you heard it.
Phagocytosis.
Autophagy. Autophagy. Self-eating.
Eating, yeah.
Autophagy. Self-eating. But that's for a good purpose. You're recycling dead organelles. Alright, so watch YouTube videos on this. And if one of them does not work for you, watch another one. Practice teaching it. Because you never know. Your name might be it. Alright?
What was the first protein?
The first protein is secreted protein. Secreted protein like hormones. Membrane proteins like sodium channels, for example. And the hydrolytic enzymes that are found in the lysosome. Alright, so all these organelles are on you. Let's switch gears to fungi. When we look at fungi here, we realize that some fungi are unicellular and some are not. The unicellular fungi is, for example, is yeast. Yeast is a unicellular fungi. And that's another thing that I need to get some students to correct some students. Again, I don't know where to get this from, but some students think that a yeast is bacteria. It is not. Please don't finish this class and go tell someone yeast is bacteria. Oh my God, boy, did I mess you up. Yeast is a fungus. It is eukaryotic. It has a nucleus, it has a mitochondria. It pretty much has a lot of things that you have. So you're not prokaryote. It is a eukaryote. It is unicellular. It is asexual. It basically divides by a process called budding. 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 Like eraser there. Budding. Let me show you. Do I have a picture of that here? Let me see. I can draw it on the board. Watch. Yeah, I had to draw it on the board. I thought I had a picture of it. All right. So budding is how a yeast cell divides. And here's what you would see, for example. Watch. One yeast cell. After a little while, a slight budding comes out of it. That's actually a new yeast cell. It is an exact copy of this one. That's why it's asexual. Now, if you fast forward in time, this might get a little bigger. And this will, of course, get a little bigger. See that? And then another one comes out. Looks like a snowman, right? Then you go a little bit more. This one will get bigger. This one will get bigger, and another one comes up. And that's how it multiplies.
So that's why it's divided or not?
That's how it divides.
Okay.
It's
So
called
it's
budding.
it's kind of its own cell division, right?
Yeah, it's
Okay.
asexual cell division. It's not
Okay.
binary fission. It's
Yeah,
called
because it's
budding.
not bacteria.
Some bacteria can do this,
Mm.
but most bacteria use binary
fission.
fission.
Yeah.
Yeast uses this.
Okay.
It would be a nightmare for you if I use this.
Mm.
Why? Because next week you'll have 3 exam lines. One from me and one from my 2 buddings. Right? So that was a bad joke. Right. Oh, I do have a picture. I was looking here and I'm like, what happened to that picture? It's right here. It's right here. I didn't see it. Did you see that? It's called a pseudohyphae.
So, quick question, so
Yeah.
when that budding system, so what happens to the other two, they kinda shed it, or?
No, they stay together.
Oh, they.
See that? Look at it. Some of them at least. Some of them stay together and they all kind of hang out with each other. You see? and it creates like a line of cells called the pseudohyphae. I'll explain to you what that is. Pseudo means false, right? It's known. Pseudo means false or not true. Not true, hyphae. Now the question is, what's a hyphae, right? Hopefully you're asking that question. Well, for me to explain to you hyphae, I gotta go to this side here. Other other fungi grow and produce what's referred to as a hyphae. This is hyphae, a network of cells attached to each other like this. That's a hyphae. And that's why they call it pseudohyphae. See, the idea is that to the untrained eye, someone who's looking at this slide, by mistake, they may call this hyphae. They may think this is this. It may look like it, but it's not it. And that's why it's called a pseudohyphae.
Not
It
hyphae.
looks like a hyphae, but it's not a true hyphae. This is a true hyphae. So the mold on your bread, when you have molded bread, that, if you look really close, you see all these, what looks like a network of fibers, and that's hyphae. Some of the hyphae is septated. That means you can see clearly where one cell begins and where one cell ends. Septated hyphae. But other types of hyphae are referred to as non-septated. That means you cannot clearly see where one cell begins and one cell ends. So there are different kinds of hyphae. But I know for a fact this one is a pseudohyphae.
What makes that pseudo?
Because
each one a cell?
It's actually a cell. The individual cells, it looks like a hyphae to the untrained eye. But to a person who knows this stuff and digs into it and understands it, goes, that's not a hyphae. Because yeast don't make hyphae. Yeast multiply by budding, and what sometimes happens is their buddings stick together. And they look like hyphae, but they're not real hyphae. So when it comes to things like molds, those are actually multicellular, microscopic, thread-like shapes from the hyphae. They can replicate asexually through mitosis and fragmentation. Meaning, this piece right here, see this piece right here? If it, for whatever reason, it breaks off and goes and lands on another part of a bread, for example, it will grow another mold in that area. That's called fragmentation. But it can also reproduce sexually, and we'll talk about that in the next slide. Now, why would any organism produce sexually? What is the purpose of sexual reproduction? Genetic variation. You're a product of that too. No two people in this room look the same. Why? Because of meiosis. Genetic variability. So the same thing here. These fungi want to also be genetically variable in case they face some kind of environmental stress, right? They don't all die. Natural selection begins to select. So you have septated hyphae, you have non-septated hyphae. Reproduction can be sexual or asexual, and so forth. Now watch this. We categorize or group fungi based on the type of spores they produce. And here are the two kinds. Watch. The sporangium is a sac-like structure. It's like a container full of spores called the sporangiospores. So that is one kind of fungi, one category. All the fungi that produce a sporangium with sporangiospores contained in the sac fall under this category. And then you got other ones, they don't have the sac. They have conidiospores, and this whole structure here is called conidium. Which one do you think is easier to flake off? The conidiospores.
This doesn't have a sac.
Doesn't have a sac. So a whole bunch of fungi fall under that category. So you got the conidium and the sporangium, the sac containing one and the non-sac containing one. All right, the reproduction, the asexual reproduction is when the spores, watch this, the spores become these hyphae cells, produce the hyphae cells. By the way, a group of hyphae cells is called the mycelium. So when the next time you look at molded bread, you most likely you can't see a hyphae because you need a microscope to see a single hyphae. What you're actually seeing is the network of hyphae. And it can dig into your bread. It can involve the entire bread, even though you don't even see it in there. It's in there. You see, some people, I don't know, they see a little green mold thing, they cut it off and they eat the rest of it, right? That hyphae is in there. Like that. Reaching out, trying to grow, eat the food. It's actually trying to eat your bread before you eat it. That's what it's doing. The network of hyphae is called the mycelium. Now, this is through asexual reproduction, mitosis. Cycle that. But then you can have a situation where there are actually two different hyphaes on the same nutrient or food. Genetically, they're different. And one of the spores, or one of these cells, flakes off. And from the other kind, and then they unite, like this. Now you have a new variety. This is sexual reproduction. You can say mom and dad, for example. And here's you. It's the same idea, right? So now you have a zygote that is genetically a mixture of mom and dad. No different than what happens with us. And they, with this new variety, creates spores, and the cycle continues. All right, fungi. Are they detrimental or are they beneficial? They're both.
Both.
Depends on your point of view. They're detrimental if you're a farmer and you wake up one day and you find your entire crop dead from a fungal infection. That's bad news for you, because there goes your money, right? So it can destroy an entire agricultural crop. They're detrimental if you're walking around and you come in contact with something. You have to come in contact with these things for you to get sick from them. So, I don't know, let's say you're walking around barefoot in the grass or something, I don't know, and you step on something, and now you have an infection. That's called community-acquired infections. When you get an infection from the
Environment.
community.
Community.
Catching it? So that means you're interacting with people, you're in the environment, you're walking around, and you happen to come in contact with someone. That makes you sick. Unfortunately, you can also get sick by going to the hospital.
Absolutely.
Wait a minute, I thought I was going to the hospital to feel better. Well, yeah. But sometimes they send you home with a present. And that's called nosocomial infections. You need to know this term, because you're going to be working in the hospital, and you need to be aware of what that is, because you're going to have to protect yourself. Because, yeah, a patient might go in for a couple of weeks or so, but you're in there for your life, for the rest of your life, working in there. So nosocomial are infections acquired during a hospital stay. MRSA, MRSA, is an example of a nosocomial infection. All right, opportunistic. Listen to this one. If your immune system is working just fine, the bacteria and the fungi within you are going to be kept in check. Your immune system is basically the guard, making sure nothing happens to you. But if your immune system weakens, this fungi that's in your gut, on your skin, in your mouth, will take advantage of that. It's kind of like a teenager. Mom and dad are not home. Yeah, let's throw a party, destroy the house. That kind of thing. So the teenager takes advantage of mom and dad, the immune system not being home, and they cause trouble. That kind of scenario, that kind of analogy. And that's called an opportunistic infection. What is the opportunity? Immune
System,
compromise.
immune weakens.
Yeah. If your immune system becomes compromised, your normal flora, which your fungi is one of them, takes advantage of that. So some people get their immune system weakens during an antibiotic therapy. So you take antibiotics, if they're strong antibiotics, your immune system will weaken to the point where you can actually get a fungal infection in your mouth. And some doctors will tell you that. You need to take this antibiotic, yeah? And if this happens to you in 2 days, this white film in your mouth, yeah, take this one. Because they know it's gonna happen. Allergies. Some people are obviously allergic to products or byproducts of fungi, for example, penicillin. Penicillin is produced by certain types of fungi. So if you're allergic to that, you'll have a detrimental consequence of that. Neurological disturbances, some of the things that a fungi releases can damage your nervous system. So all of these are referred to as detrimental. Is there anything good about them? Here it is. We talked about the fact that they're saprobes. Do you remember what that is? What is it?
Viruses?
Say again?
Virus?
They're recycled.
Recyclers. Saprobes are recyclers. I just mentioned penicillin. So we can get antibiotics from them, right? Penicillin. My favorite one.
Heineken.
Yeah, there you go. Make beer, vitamins, food flavorings. For example, blue cheese. I don't know if you like blue cheese or not. Anyone like blue cheese? Most people like it.
The stinkier the better.
Oh yeah, blue cheese is awesome. You guys you don't know what you're missing. And you're probably like, fungus on my food? That's disgusting. Hey, can you pass me that mushroom pizza? You catching me? Mushroom pizza, if you eat mushroom pizza, that's a fungus. But blue cheese, no way. Come on, it's the same thing. Actually, blue cheese is very expensive. The right kind, not the standard blue cheese that we see in the supermarket here. But the right kind is not cheap at all. All right? Making bread. How do you think your bread rises, right? It is basically because of the anaerobic, the fact that yeast is facultative. You can basically have it do anaerobic cellular respiration or aerobic cellular respiration. So a byproduct of yeast fermenting is alcohol. Another byproduct of yeast fermenting is carbon dioxide. And carbon dioxide is what allows the bread to rise.
I make challah bread from scratch. It's like a 7-hour process, but it's always fun to see, like when I mix the yeast with a little bit of sugar in the warm water.
It starts
and
bubbling.
Within minutes it starts bubbling.
Yeah.
And then when you let it sit, like 2-3 hours later, it's like it's always fun.
Yeah, I want to see if I can start doing those things. I tell my wife, I want to start baking bread. She goes, oh no.
It's the only one
She's
I
like,
make.
oh no. You don't need it. Anyways. All right, protozoa or protists. These guys are your unicellular organisms that live in fresh water. These are the protozoa. The algae are photosynthetic. They are unicellular, some of them, but some of them are also multicellular. So this is a nice handy table that gives you the similarities and the differences between the two. Now they both fall under protista. They're both protists. Do not confuse the word protist with protozoa, all right? Why did I tell you this? Because I used to confuse it when I was a student. But protists is the umbrella that has algae and protozoa under it. Protists encompasses all of the algae and all of the protozoa. So the protozoa, they're only unicellular, but the algae, you have examples of both. With respect to photosynthesis, only algae. Now, there are some members of the protozoa, they have no idea what to do with them. Do we put them here under protozoa, or do we put them here under algae? Why? Because they have features of this and features of that. So there are some places that say, well, for this one, you say either one. It's fine. Because they don't know what to do with it. An example of that is something called euglena. They don't know what to do with it. It's a protozoa, but it's got chloroplasts. So I think they need some kind of update. Maybe another category, maybe something like that encompasses both characteristics. All right, photosynthetic algae, but for the most part, only algae is photosynthetic. These guys are not. Then you got autotrophic. Well, if you're photosynthetic, you're autotrophic. If you're photosynthetic, that means you're making your own food.
Yes.
And that's what autotrophic means. If you're not photosynthetic, then you're heterotrophic. That means you have to eat food. Which one do you think we are? Heterotrophic. Yeah. And because protozoa, listen to this one, because protozoa are heterotrophic, we can now put them with the saprobes. Remember which two are saprobes? Bacteria and fungi. There's another one, protozoa. We're adding protozoa to it. They can recycle things too. Where do they live? A lot of similar places. A lot of overlap here. I'm not very big on asking that kind of question, but I just wanted to be thorough. Look, they live in very similar places. All right, their essential role or benefits. Well, algae, essential role, oxygen. 70% of the oxygen that you're breathing right now is from algae and photosynthetic bacteria. So that is an essential role. But there's another essential role that escapes a lot of students. Plankton. The aquatic food web. That means the food in the water. It begins at plankton. Plankton basically is a community of microorganisms, mainly algae. But it's got other things. We'll have protozoa in there, bacteria in there, other things. And they're all swimming together on the surface of the water, you see? And who's eating them? Not me. The little tiny fish. Now watch this. Let me try to catch how many tinies I say. The little tiny fish eat the plankton. Sounds good? Mm-hmm. Then the tiny fish eat the tiny fish. You catching it? And then the tiny fish eat the tiny fish. You catching all that? Eventually you get the sharks and so without plankton, there would be no sea life. And with no sea life, we wouldn't be here. Look how crucial and how fragile life is. On the other side, protozoa's essential role is the fact that they're recyclers, alongside with fungi and bacteria. Environmental concern. There's no environmental concern with the protozoa. With algae, it's basically their secretions is what's concerning. Have you ever heard of red tide? Down south, towards end of July, early August, when the weather
Mhm.
is exactly perfect there, you get what's called an algal bloom, where the contamination of the water, all that. It's just perfect environment to get the algae to bloom, meaning they multiply way out of control. Well, the algae can secrete toxins into the water. That's what the concern is. I'm not really specifically concerned about the algae. I'm more concerned about their secretions. That's what's going to hurt you. So obviously they put flags up, no swimming, no fishing around that time. So if you eat fish that swim in this kind of water, that is, of course, would be harmful to you. So you have to watch out during that time of year. All right, when it comes to algae and disease, watch this. Algae will not harm you. Algae itself will not harm you. It's their secretions that harm you, the toxins. If you ingest algae, the algae itself it's photosynthetic. It's gonna die. There's no sun inside. So it gets no benefit out of you. But their secretions are harmful. So you don't want the toxins in you. Until you get to this one. Watch this. You can get this by, if you dig in soil and it happens to be there, you can get something called protothiocosis. This is not your average algae. Listen to me. This is a mutated algae. And when it got mutated, what happened to it? It lost its photosynthetic capabilities. This algae is no longer photosynthetic. So now what does it want? It wants food from you. You come in contact with this one, you will get sick. That's the whole idea. Why does bacteria hurt you or whatever? Well, they're taking food from you and multiplying, secreting things. So you're going to get sick from that. Algae, your standard algae, will not harm you because they don't need anything from you. They're photosynthetic. Unless you come in contact with this one, the mutated one, and it's in soil. You get what looks like skin rash, and it looks like you have chickenpox kind of thing. So why does it hurt you? Why does this one hurt you? Because it lost its ability to photosynthesize. Now, for it to survive, it has to basically take things from you. And when it does, it hurts you. When it comes to protozoa, there are a whole bunch of them that are harmful to you. Let me give you examples of this. Malaria is the plasmodium. This is transmitted through the bite of a mosquito. And I think I have some of these, if not all of these, in the disease connection maps. The mosquito that's harboring the plasmodium protozoan bites you. And here's the interesting thing, watch. The plasmodium runs and hides in your liver. What is it trying to do? Avoid your immune system. It goes inside your liver cell, trying to get me now. For you to get me now, you got to kill your liver cell. So your immune system, hang on. Hold it, wait. We'll just wait out here. Meanwhile, the plasmodium is multiplying inside your liver cell. One goes in, 10,000 come out. And when they come out, they come out from the back door. Why? Because they know the immune system is at the front door. And they come out and they run and hide in your red blood cells. And that's basically what malaria is. Malaria, now you will have the plasmodium in your blood cells, red blood cells. Why? Why the red blood cells? It's amazing if you think about it. Who came to bite you? A mosquito.
Mm-hmm.
The mosquito came to bite you, introduced plasmodium, right? The plasmodium goes, multiplies. It's got to leave. It wants to leave, go home. It wants another mosquito to come and bite you again to take it back out. Do you see what I mean? And that's why it hides in your red blood cells. Because where are your blood? They're right there, everywhere. it's increasing its chance or opportunity to leave your body. So it's waiting for the next mosquito to bite you, take a little bit of blood with it, and now it's leaving. It's fascinating.
And go give it to someone.
Obviously, meanwhile, you are sick. You have to obviously take medication. African trypanosomiasis, say that 10 times, right? African trypanosomiasis is caused by Trypanosoma brucei. This is basically sleeping sickness. This, when it is transmitted into you, this is the tsetse fly. I love saying that. The tsetse fly comes and bites you, transmits the Trypanosoma brucei. The initial symptoms is flu-like until they eventually reach your brain. You will start losing your sleeping cycle, something called circadian rhythm. 2 o'clock in the morning, you're not sleeping. 4 o'clock, you're still not sleeping. Right? So it's called sleeping sickness because your sleeping cycle gets all messed up. But by then, oh, you are in trouble. You need medical attention because now it's in your brain. The problem with this is some place like here, United States, can be easily overlooked as the flu. But in certain parts of the world, they are aware of it, where the fly is, so they know to at least take a look and make sure that it's not this. Giardiasis causes basically diarrhea. Toxoplasmosis, listen to this story. Anybody has cats at home? Cats? Do they leave the house? Does anyone have a cat that leaves the house? Wanders the neighborhood? I don't have cats, but I have a cat that lives nearby that every time I go to work and here she is. Look at it. Who owns this cat? Don't they know about this? I'm trying to find out who owns this cat. And I'm going to give him this lie. Listen to this one. If you have a cat that wanders the neighborhood, watch out. Does this cat come and tell you, yeah, when I was out, yeah, I ate 4 rats, ran after 5 mice, I didn't know. It's just going to come back. I'm back. You run the risk of having a cat come home with toxoplasmosis, with the Toxoplasma gondii. That's the protozoa. What is this thing? Watch this. This thing is amazing. The Toxoplasma gondii wants to enter the cat as the host. And it knows that the rat, R-A-T, is afraid of the cat, C-A-T. So look what it does. It infects the rat, R-A-T. And it changes the rat's behavior. It manipulates the rat's behavior. What does it do? It tells the rat, hey, you know the cat, yeah? I know you're usually afraid of the cat. And I know you usually run away from the cat. But I'm going to change your mind. The cat is your friend. The next time a cat comes nearby, yeah, don't run.
Go say hi.
Go say hi. I like that. Why? Because the Toxoplasma wants the cat to eat the rat.
So that he can get it.
It's amazing. The protozoa actually affects the behavior of the rat so that it is no longer fearing the cat. And sure enough, it becomes lunch. But now where's the Toxoplasma?
In the cat.
It's in the cat. And that's where it wants to be. That's amazing. But now that cat comes home. It goes into your litter box. And now it's shedding the Toxoplasma in your litter box. And if you go change the litter box without gloves, a mask, or whatever, now you have it. Now, one interesting thing is this. At most, you will get flu-like symptoms. At most. So they're not really, it's better not to get it. But at most, unless you're pregnant. If you are pregnant and you have a cat that leaves the house, do not touch the litter box. Let your husband do that. Or your boyfriend, whatever. Don't come near it.
Near the cat?
Near the litter box. Don't even try it. All right? Just in case. I'm talking about a cat that leaves the house. If the cat isn't home, you don't have to worry about it. The idea is this. If a pregnant woman contracts this protozoan, the problem is not, it's not going to hurt the woman, it's going to hurt the baby. That baby will be born with birth defects. So obviously you want to be aware of that. This disease here at Chagas, it comes to you with this is Trypanosoma cruzi. It comes from the bite of one of the ugliest things you'll see. During the break, I'll see if I can pull up a picture for you. It is called, the insect is called Trypanosoma, not Trypanosoma. It is something infestans, I can't remember. I will look it up. But anyways, this guy here comes to you, the insect that bites you has this in it, and it comes to you at 3 a.m. What does it want? It wants to kiss you. It's called the kissing bug. It comes to you when you're sleeping, you're not moving, you're not aware of what's going on. And it comes near your mouth and it kisses you. It's not kissing you. It's defecating on you. And when it defecates, it releases this guy, and you feel something when you're sleeping, so you go like this. Now it's on your hand.
You're not making this class fun anymore.
I know. Now it's on your hand. You're not aware of what happened, and eventually you will contract this. Here's the interesting thing. Nothing happens to you. For most people, nothing happens. I think I remember reading 60 to 70%, it doesn't do anything. But if you're one of the 30%, nothing happens to you. Even if you're one of the 30%, nothing happens to you for 20 years. 20 years later, you'll have congestive heart failure and a whole bunch of other things. Now the problem is, did you get congestive heart failure because you got old? Or you're older? Or did you get it because of this? Unless you test for it, you wouldn't know. So you'd walk around for 20 years with it, not knowing it. Unless somebody happens to look and find, oh my God, look what he's got. That's a different story. But a lot of people can walk around, nothing happens, or nothing happens for a while, and it all hits all of a sudden. Balantidiasis. This is the only ciliated protozoan that causes disease. This is a protozoa that has cilia, and I will talk about it, that causes disease. This one causes explosive diarrhea. Explosive. What does that mean? That means your colon blows up. You don't survive that. I mean, these things they're like very harmful. And then you got this one here, brain-eating amoeba, Naegleria fowleri. You have to worry about this one in Florida. Naegleria fowleri multiplies much quicker during hot weather, temperature, hot water. I'm not talking about warm water. I'm talking about water that you go in and swimming, for example, end of July, early August, where you say, oh my God, it's so good, it's so warm. Get out. Because if you like it, they like it too. This guy can gain access to your brain through your sinus. It bores a hole into your brain and it goes, brain tissue. And it eats your brain. You don't survive that.
How does it get through the blood-brain barrier?
It eats through the tissue. So it's not going through your blood, it's going through your sinus. So when you go swimming, water will enter your nose, whether you try to avoid it or not. And if it happens to have one droplet, happen to have this guy, it's gonna bore through your sinuses, very easy access to your brain, and it just eats brain tissue. That's why it's called the brain-eating amoeba. Yeah?
Okay, going back to the toxoplasma, what's the difference between a cat bringing that into the house and you going outside where rats have been and inhaling? Like, if it's in a rat and they're outside, can't you still get it?
Well, the rat might have to bite you or something, or you may have to eat it. You see what I mean? But with the cat, is an easy transport into your house through the cat's fecal material in the litter box, and then you're mixing when you're taking the litter box out to clean it.
So it's not just
It's up in the air,
airborne.
huh?
It's not just airborne. It has
No,
to
you
be-
have to come in contact with it. Yeah.
For that same one, like the same way it affects like behavior in the rat doesn't affect the behavior of the cat.
Beautiful question. We don't know. I don't care about the cat. Right? The cat will behave the way they behave. Right? I worry about the human who got it,
Yeah.
and their study is being done on that. Does that affect the behavior of a human being that contracts it? The answer to that is still up. We don't know yet. Let me quickly talk about this and I'll have this done. I'll give you a good, nice break. This is how protozoans move. So if you're a protozoan and you have cilia, you fall under the category called ciliates. How easy is that?
Yeah.
If you're a protozoan and you have flagella, what do you think?
Flagellates.
Flagellates. So I'm done. You agree? The cilia are ciliates, the flagellates are flagellates. Okay. The only two I really need to study is this one, because nothing in that word tells me pseudopodia. Sarcodines. Nothing in that word tells me that's a false foot. So amoeba are sarcodines, pseudopodia. They use false feet to move. This one is even worse because sporo implies spores. Am I right? Nothing to do with spores here. Sporozoan means no means of motility. Sporozoan means I don't have any of these. That's what it means. So don't get confused with that word. Because I know, again, I would think it means something that has to do with spores. Not in this case. Sounds good? Real quick. This you already know. Believe it or not. You already saw this. When we talked about bacteria endospores, remember that?
Mm-hmm.
The bacterial endospores under harsh conditions, right?
Mm-hmm.
The vegetative cell, bacteria, produces an endospore under harsh conditions. Am I right or not?
Mm-hmm.
And then the endospore, once the environment is back to normal, it germinates. Am I right?
Mm-hmm.
No difference. It's the same exact thing. Under harsh conditions, they also do this, but the names are different. Instead of the bacterial vegetative cell, we call it the protozoan's trophozyte. The metabolically active protozoan is a trophozyte. So you replace the vegetative bacteria with trophozyte protozoa. Sounds good? When the trophozyte experiences harsh conditions, it's not going to produce an endospore. It's going to produce a cyst. So instead of endospore, it's cyst. But the idea is exactly the same. Good, let's take a break. 5-minute break. Well deserved. Almost done with it.